All right, today we're going to continue our discussion of bioenergetics and metabolism, looking at ATP. So our learning objective this time around is to summarize the role of ATP in cell physiology. Now, here is our ATP. Um, this again is a nucleotide, so we have our nitrogenous base, we have our ribose, and attached to that is three phosphate groups. Triphosphate, 1, 2, 3. Now, when we hydrolyze off phosphates, we can achieve this in one of two ways. We can hydrolyze off one phosphate group at the end, leaving a DP, or diphosphate, and a single inorganic molecule. This releases a certain amount of energy. That energy is gleaned both from the increase in entropy by now having two molecules as opposed to one, but also we get a release of potential energy that is stored in the bonds holding these phosphate groups together. Keeping in mind that these phosphate groups are very electronegative, they tend to be repelling each other, and that bond is essentially holding a lot of potential energy. As soon as we hydrolyze it, that energy is released. It now becomes kinetic energy that can be used to do work. Similarly, instead of taking off a single phosphate, we can take off two phosphates, leaving only a MP, a single monophosphate group, but also liberating this pyrophosphate compound, which is two inorganic phosphates still coupled together. Pyrophosphate inside of a cell is rapidly broken down into individual phosphate molecules. So here we actually get release of energy from the potential bond and increase in entropy here, and another boost over here. So ATP has a lot of potential energy and a lot of possibility for increasing entropy. So consequently, it is inherently has a negative delta G. So how does that help us do things? Well, if we have a particular reaction, such as A becoming B, and it has a positive delta G, which means that it is an unfavorable reaction, if we can somehow couple that to a negative delta G reaction, consequently, an example of this would having the same enzyme carrying out this reaction also carrying out this reaction simultaneously, the net effect of both of them is to have a net negative 20 delta G. So here we've essentially harvested the energy to do this positive delta G reaction by also doing a high negative delta G reaction and still having negative delta G left over. Now, you might ask yourself the question, why ATP? Why not GTP or CTP or UDP or any of the others? Because there really is no difference, all right? They all have the same amount of energy in those triphosphate groups. This is kind of a two-pronged aspect. So the reason why ATP was picked is it tends to be more abundant in the cell, and as you'll see later, the adenine nucleotide is actually a little bit more difficult to make than CTP or TTP or UTP. So ATP and GTP are a little bit more difficult to make, so we would have a tendency of wanting to retain that in the cell. one way of retaining it is to utilize it for other things other than just as a nucleotide, right? So here's one driving factor selecting, in this case, either ATP or GTP. The secondary thing is just a consequence of evolution. So at some point in time, an enzyme developed an ATP binding site that worked very well, and the end result of that is that binding site was replicated into other genes, as one gene became another gene or became another gene. As evolution occurred, it retained that binding site. So remember that, when we talk about evolution, not only are we talking about one organism changing a little bit over time to become something else, so consequently you can have a whole slew of different birds evolving from a single bird, but this also works down at the gene level, right? So we can have a single gene that gets duplicated and starts changing or evolving, and down the line, you end up with multiple genes that are all evolved from a single source. So having a good ATP binding site a long time ago means that it's evolutionarily favored. So that's really the only reason. Um, Consequently, since we have this in the cell and it is a high negative delta G, we can utilize it as an energy currency, right? So we can store this up, we can keep it, we can use it when we need to, but sitting around by itself, it is in itself fairly stable, right? This is in contrast to something like NADH, which we'll see as being one of our universal electron carriers, that is inherently reactive and can cause problems. Having a bunch of ATP around is not going to cause those issues. And we'll go into more detail about that later on. Again, a lot of that energy comes from the large amount of electrostatic repulsion of the negative charges, but also has a lot to do with entropy. Now, if we take a look at the actual chemical reaction when we talk about hydrolysis, what you'll see here is that a water molecule, a lone pair from the central oxygen, is going to act as a nucleophile attacking that phosphate, and it just so happens that this oxygen is going to be the best leaving group, and those electrons, since we're forming a new bond here, we're going to lose one bond. Those electrons are going to come back in. This will turn into a double bond, which will kick this double bond out into a single bond. So if we look at this terminal phosphate, any one of these three oxygens could carry the double bond, and it's flexible, it's floppable. So consequently, when we attack this with a pair of lone electrons, this is a better leaving group. Those electrons come over here, and these electrons come back out. Again, when we go to attack the next one over, it's essentially the same reaction. We attack in here. This is a better leaving group. The electrons come form a double bond here. These get kicked out into a single bond. And same thing happens over here. So you can see that the first two hydrolysis reactions are very high energy in terms of having a large negative delta G. The last phosphate removal is still a negative delta G, but has a lot less energy. Notice here that a single phosphate group is not in tension with all of these other electronegative groups. So here we have two phosphates pushing each other away. Here we have three phosphates pushing each other away. That adds to the larger amount of negative delta G. So that last phosphate hydrolyzation is not really functionally significant. So we'll see one coming off or two coming off, but typically that last phosphate just stays put. So we discussed the role of ATP in cell physiology.